terminal oxidation or oxidative phosphorylation. So what is meant by this terminal oxidation? Otherwise also we termed it as electron transport system, ETS, electron. Okay, so reaction, metabolic process occurring on the inner membrane of the mitochondria. Okay, so it's a last step of aerobic respiration. Terminal oxidation is the last step of aerobic respiration. What is its significance? For what purpose this reaction is going on? So we can see. Here in aerobic respiration there are mainly 3 different steps are there. First step is glycolysis, second Krebs cycle like that, and third one is terminal oxidation. So these are the 3 steps. We know that already. Glycolysis, the first step in which the glucose undergo partial oxidation. So here as a result we know that the cell can produce 2 ATP and 2 NADH2 from this process inside the cytoplasm. Okay, so then after that the pyruvic acid, 2 molecules of pyruvic acid, then from 1 glucose molecule then enter into the mitochondria. Okay, so inside the mitochondria it undergo further oxidation. Okay, and as a result the 8 molecules of NADH2, 2 molecules of 2 FADH2 and 2 GTP molecules are also produced inside the mitochondria. Thus the pyruvic acid is completely oxidized. That is what we studied in Krebs cycle. So these are the 2 previous steps. Okay, so we know that the cell needs energy. Okay, so for that purpose ATP can act as the energy currency. Wherever there is energy requirement, so cell can take the ATP for that purpose. Okay, so NADH2 molecules are also energy rich compounds, but cell cannot take it directly, okay, as a source of energy. Okay, so NADH2 molecules we know that so one NADH2 can carry an energy equal to 3 ATP. Okay, similarly one FADH2 molecule having an energy equal to 2 ATP. Okay, so these molecules need to be converted to ATP, then only the cell can use that energy. Okay, so these reduced enzyme need to be oxidized. Okay, and the energy need to be stored in ATP. Thus the cell then the cell can use that energy for the metabolic process. Okay, so that reaction is the terminal oxidation. What is that? The oxidation of which all compounds? Oxidation of NADH2 and FADH2, and results in the formation of energy rich ATP molecules. So that process we can term it as terminal oxidation. Okay, so that terminal oxidation occur on the inner membrane of the mitochondria. Okay, so here we know that the mitochondria there are 2 membranes. So this is outer membrane, then inner membrane is there in the mitochondria. So between that there is a space is there. We can term it as perimitochondrial space, PMS, intermembrane space or perimitochondrial space. So and at the center there is the matrix of the mitochondria. We know that. So this is the central fluid filled region, cavity of the mitochondria. So where the Krebs cycle is taking place inside the matrix, you know that. Okay, so here inside that matrix large number of what is that energy rich reduced coenzymes are there. NADH2, FADH2 like molecules can be seen inside that region, okay, matrix. So these molecules need to be further oxidized. Okay, so so that reaction occur on this membrane. So this is the inner membrane, okay, where the further reaction occur, oxidation and reduction. Okay, so here that membrane inner, for that purpose the inner membrane carries some electron carriers, something is there. Okay, here that diagram or picture is somewhat enlarged here. Here you can see. So here this is the outer membrane. So here you can see. So this is the outer membrane. I just shown like this. So this is the outer membrane of mitochondria, and this region is the inner membrane of the mitochondria. IEM, inner membrane region, and the space in between we can term it as intermembrane space. Okay, here can be seen, and inside that here the inner fluid filled region cavity, that is what we termed it as matrix of the mitochondria. Okay, so here the energy rich coenzymes like NADH2, FADH2 like molecules, coenzymes can be seen inside the fluid matrix matrix of the mitochondria. So these energy rich coenzymes are produced through Krebs cycle. Okay, you know that. So these compounds, molecules can be seen here. Okay, so the reaction that we are going to discuss usually associated with this inner membrane. So here that inner membrane contains some electron carriers are there. Inner membrane carries some electron carriers for that purpose. Okay, electron carrying elements are there. So the electron carriers can be arranged in 4 complexes. Okay, electron carriers are arranged in some units known as electron carrier complexes. Okay, right. So here there are we can see there are 5 complexes electron for that purpose. There are 5 complexes can be seen on the inner membrane. So here we can start from here. So that complexes are responsible for the oxidation of these molecules. Okay, so here the complex 1 we can see here, complex 1 here otherwise we named it as NADH dehydrogenase complex. Okay, so that is the name of this complex. So it having 2 members are there. The complex 1 contain 2 members, FMN and FES. So these are the components of this complex 1. So the complex 1 having 2 elements or members are there, FMN and FES. FMN is actually flavin mononucleotide. Okay, and the other one is an iron sulfur protein. Okay, so they together form this unit. They are responsible for the oxidation of NADH2. Responsible for the oxidation of this NADH2. Okay, so this NADH2 molecule undergo oxidation. That means electrons are removed from this molecule. 2 electrons are removed from this molecule. So that electron is taken away by this complex 1. Okay, it oxidizes this complex 1. Otherwise we can say oxidize the NADH2 and remove the electrons. Okay, okay. So along with that the 2 hydrogen ions are also removed from NADH2. 2 hydrogen ions and the other 2 electrons are removed from NADH2, and this hydrogen ion is transferred to the this outer cavity. Okay, 2 hydrogen ions are transferred to the outer cavity. So that is outer cavity. Okay, and then the electrons passes through these carriers, and then it is given to another molecule. So that is known as ubiquinone. So here complex 3 is here, complex 3. Complex 3 receive the electrons and hydrogen ions from ubiquinone. Okay, and these electrons are transferred or passed among them. Inside that complex 3 there are two components are there, cytochrome B and cytochrome C1, and one FES is also there. So the electron is passed here, and along with that the hydrogen ions are pumped to the outside cavity, perimitochondrial space. The hydrogen ions are pumped to the outside, and the electrons are electron is passed inside the complex 3, and what will happen ultimately, the electron is given to cytochrome C. Is another electron carrier, not a complex. That carrier can be seen here on the outer side of the inner membrane. So this is the inner membrane, you know that, and this cytochrome C can be seen on the outer surface of the inner membrane, attached to the outer side of the inner membrane. So here the cytochrome C is an important component, and here it is a mobile electron carrier. Cytochrome C is a mobile electron carrier. So here you can see, and it carry this electron from here, from where, from complex 3, and it can move forward. It is a mobile carrier, and it move downwards, and then what will happen, it ultimately reach the complex 4 here. Cytochrome C reach near the complex 4 and released or donate that electron to complex 4. Okay, so here the complex 4, here we can see is, otherwise we named it a cytochrome C oxidase complex. Okay, cytochrome C oxidase complex. Here there are two members are here inside that complex 4, cytochrome A and A3. So they are the members of this complex 4. So here there are two copper atoms. Copper atoms are also there for the oxidation reduction purpose. Okay, with the help of these elements they can receive the electrons. Okay, and while the electron is passed within the complex 4, okay, then two hydrogen ions again here can be pumped from inside to the outside, from inner matrix of the matrix to the outside. To here again two hydrogen ions are pumped to the outside. Okay, so then by using the flow of the, by using the energy of this electron here. Okay, so the two electrons are then ultimately given to oxygen. Okay, taken or received by the oxygen. Two electrons combine with the oxygen, and along with the two hydrogen ions also we can see attached to it. Thus that lead to the formation of water molecule. That lead to the formation of water molecule here. So here here the role of the oxygen can be seen here. In aerobic respiration the oxygen can be seen here as a final electron acceptor. Finally electron is received or accepted by the oxygen. Otherwise sometimes we are also be termed it as final hydrogen acceptor also. Here this is the role played by the oxygen here in respiration. Okay, so that leads to the formation of water molecule. Okay, so we can see here the flow of electrons occur. We can see as a result of the flow of electrons, what happened here. So here hydrogen ions are pumped to the outside at different places. Okay, by using the energy of the electron. Okay, so by using the energy of the electron. Okay, so here two hydrogen ions are pumped to the outside, where in the first complex 1, and here we can see another pair of electrons pumped to the outside here, and again two pairs of hydrogen ions here also. So 3 pairs of hydrogen ion, 6 hydrogen ion we can see moved to the outside. Okay, here how that movement occur? By using the energy of the electrons, okay, obtained from the oxidation of this NADH2. Okay, okay, yes. Then what will happen as a result? So the continuously that process is going on. So NADH2 oxidation, the oxidation process continue. So then what will happen? The hydrogen ion concentration increases in the outer compartment, and it decreases in the inner compartment. So the concentration of the hydrogen ion gradually increases here in the outer cavity. As a result, the hydrogen ions develop a tendency to flow back into the matrix. A proton gradient develop. Here a high concentration of protein, and here there is a low concentration of protein. So the protein need to flow back into the matrix. So there is a reverse flow can be seen. Okay, that movement of proton back into the matrix occur through a special type of protein complex present in the inner membrane. So that is what we can term it as oxisome of FOF1 particles. FOF1 particle appear like this. You can see here this is the structure of that particle, and there is a this particle is a protein molecule having here there is a base and a head region is there, can be divided into two, base and head. Base region can also be termed it as FO, and the other one is known as F1 particle. FO and F1 particle. Base is otherwise also known as proton channel. Base also known as proton channel. Okay, and the head can also be termed it as ATP synthase and same. Synthase and same. So that is we can say, see here that molecule is very important here again. So FOF1 particle here play an important role. So we can term it as complex 5. So this molecule act as the complex 5. So this is not a electron carrier. So here hydrogen ions, so here FO particle, so the base region act as the proton channel for the facilitated diffusion of proton from outer space to inner space. So through the FO region, the proton can easily enter into the matrix region. So here in the other regions, here in this region, the proton cannot flow through this region. Okay, so the movement of the proton can occur only through this region. Okay, into the inner compartment. So that is the importance. So here when the proton flow occur through this FO particle, FO is the act as the channel for the movement of the proton. It help in the facilitated diffusion of proton from the outside to inside. So from the outer compartment, the proton flow with a very high speed here. So by using the energy of that reverse flow, what will happen? ATP will be produced from ADP. Okay, so in the NCERT text it is given like this. The F1 particle, the head region, develop a structural change, we can say, so that the F1 particle, otherwise we termed it as ATP synthase enzyme. ATP synthase enzyme. So that enzyme develop a conformational change that lead to the formation of ATP from ADP. So here ADP and inorganic phosphate combine together and ATP will be produced. Okay, so that means when the proton flow occur, the ATP synthase enzyme get activated. So that lead to the formation of ATP. This F1 head piece become functional or activated when the whenever there is a flow of protons. When the flow of protons occur through here, the head region get activated, or there occur a structural change or change here in that region, in that enzyme, that can lead to the formation of ATP from ADP and inorganic phosphate. So that is what we can see here. When the flow of protons stop, then what will happen? The head region or ATP synthase enzyme get inactivated. So the ATP synthesis stop. Okay, so that is what we can see here. So here the flow of 2 hydrogen ions is required for the synthesis of 1 ATP according to NCERT. In the new NCERT it is given little bit different. Flow of 2 hydrogen ion is required for the synthesis of 1 ATP. At least 2 hydrogen ions needed for the formation of 1 ATP. Okay, so as a result of the oxidation of NADH2, how many? 3 pairs of hydrogen ion. One here, pair here, here again, and here again. 3 pairs of hydrogen ions are pumped from the inner space to outer space. So that can lead to the formation of 3 ATP. So 1 NADH2 can lead to the formation of 3 ATP. So that means there are 3 pairs of hydrogen ions pumped to the outside. So that means that can produce 3 ATP. Okay. Yes. Till now we discussed only this complex 1, 3, 4 and 5. Complex 2 is also there. What is the role of complex 2? Okay, complex 2 I mentioned here. This is the complex 2. Okay, so complex 2 is succinate dehydrogenase complex. So that is the name of the complex, succinate dehydrogenase complex. So here that complex here is responsible for the conversion of succinic acid to fumaric acid. Okay, so that it acts as the enzyme for the for one step of glycolysis, Krebs cycle. Okay, succinate dehydrogenase, that enzyme is present on the inner membrane of the mitochondria associated with the complex 2. Okay, so that succinate dehydrogenase, while converting succinic acid to fumaric acid, okay, what will happen? FADH2 will be produced. Okay, FAD is present inside the complex 2. The FAD during that reaction, FAD receive the hydrogen ion and electrons from succinic acid. Okay, in Krebs cycle, from succinic acid these electron and hydrogen ions are received by FADH2 and it can convert it to fumaric acid. Okay, so the FADH2 is the complex 2. From FADH2 FADH2 is produced. The complex 2 can also contain FES. So here from the electrons are transferred to ubiquinone. Here from the complex 2 also the electrons and hydrogen ions are transferred to ubiquinone. Here again we can see ubiquinone here. Here also ubiquinone is there. So the ubiquinone receive the this reducing power and get converted into UQH2. Okay, UQH2, right? Reduced ubiquinone is known as ubiquinone. Okay, right? UQH2 is produced here, and this UQH2 release the ubiquinone, release the donate the electrons to again complex 3. Okay, complex 3. From here also the electron is given to complex 3. So ubiquinone acts as a electron carrier between complex 1 and 3 and between 2 and 3. Okay, from here electrons transferred to complex 3, and from here also electrons are transferred to complex 3 with the help of the ubiquinone itself. Okay, so here electron received by the complex 3, and then what will happen? The hydrogen ions are here transferred to outer compartment. 2 hydrogen ions present here in the ubiquinone, again here from here also transferred to the outside. 2 hydrogen ions can be seen here, pumped to the outside, and the electron passes within the complex 3 and at last given to the cytochrome C, similar to the previous case. Okay, so here again cytochrome C receive the electrons from complex 3, and again the same process occur. The cytochrome C move towards the complex 4, and the electrons are given to complex 4. The same process occur. Then during the movement of the proton, electrons here within the complex 4, again 2 pairs of hydrogen ions are passed to the outside, pumped to the outside. Okay, and here then the electrons are given to oxygen, and that lead to the formation of water molecule. Okay, so that is another movement. Okay, so here the electron movement occur from here, from succinic acid, like that, from complex 2 to complex 3, and then cytochrome C, and then come to the through complex 4 to oxygen. So that will happen. That happened like this. So as a result of this second flow of electrons, again 2 pairs of hydrogen ions are pumped to the outside. Okay, here 1 pair hydrogen ion, and here 1 pair hydrogen ion. Okay, so here who is oxidized here? FADH2, by through the oxidation of FADH2. So from here from FADH2 hydrogen ion movement occur, electron movement occur. So as a result, 2 pairs of hydrogen ions are pumped to the outside. So as we discussed earlier, 1 pair of hydrogen ion is required for the formation of 1 ATP. So here 2 pairs of hydrogen ions are there. You can see 2 pairs of hydrogen ions are there. 1 pair this one, and the other pair this one. So that can produce 2 ATP. 1 FADH2 can produce 2 ATP. So that is what we studied earlier. Okay, so this is the overall process of terminal oxidation or electron transport system. But here in our new NCERT text, it is given like this. Here what is that difference we can see? Passage of for each ATP produced, 4 hydrogen ions passes through FO from the intermembrane space to the matrix. According to the new NCERT, it is given slight difference is there. So you need to be careful. 4 hydrogen ions need to be passes through the F1 particle for the formation of 1 ATP. So that is what mentioned here, not 2 hydrogen ions. Okay, so slight difference is here. You need to be careful. So that is the overall process. So the terminal oxidation. So here the oxidation reduction process here on the inner membrane responsible for the formation of ATP. So the oxidative phosphorylation. So this type of ATP synthesis we can term it as oxidative phosphorylation. Okay, so that oxidation reduction. So oxidation of the reduced coenzymes lead to the formation of ATP. Okay, terminal oxidation. So that is all about that process. Okay, thank you.